Chapter 10, Chapter 11. Chapter 10 is about vision, and Chapter 11 is about muscle movement. I'm not exactly sure why I put those two together, but let's go ahead and get started. There we go. Chapter 10, vision. The whole area that you can see without moving your head or eyes is known as your visual field. While the visual field may be wide, an individual's visual acuity tends to be only in the middle of the field of vision. This is because there is a concentration of cones in the middle of your visual field, an area known as the fovea. So you don't see as well out here. Here I am trying to show you. You see better in the middle of the field, and that's one of the reasons I was thinking of getting glasses. I have presbyopia, which is old eyes. Presby means old. I have presbyopia, and my readers are about 2.5 times stronger than just my vision. So I've got them on right now. That's the glasses you saw me wearing. They were readers. But when I went to the optometrist and they made glasses for me, that was the only focus there was right in the middle of the field. And everything else was blurry. And it was terrible. I couldn't see. My peripheral vision is really, really good. So I couldn't see. And so I didn't buy the glasses. And they got upset. But I guess I go back and, I don't know, and next week sometime to get my eyes checked again. They'll try to convince me to get glasses. Anyway, so you usually look directly at something. That's where your phobia is. Even with a concentration of visual receptors in the center of the visual field, there is a spot where the optic nerve feeds out of the eye and blood vessels feed into the eye. This is known as the optic disc. And this area maintains a blind spot in an individual's visual field. Normally, the brain automatically fills in the blind spot, so you don't realize that it's there, but you do have a blind spot. But your brain is thinking, well, I'll just fill it in with whatever's around it. And that's what you got. Vision comes to humans in the form of wavelengths of light or photons. The human visual ability lies between 400 and 700 nanometers. That's ultraviolet to infrared light. Both ends look black. Other animals have ranges of vision that encompass wider spectrums of light. Bees can detect beyond ultraviolet light. A photon of light comes into your eye with select properties that we will perceive as color. The hue is the wavelength of the light. Wavelengths in the 400 to 500 range are very dark purples to blue. Wavelengths in the 500 to 600 range are greens to orange, with yellow in the middle. Wavelengths in the 600 to 700 range are bright red, darkening to black. Since most color is detected as patterns of light of different wavelengths, brightness refers to the amount of white light from the object. Saturation has to do with the amount of light of a select wavelength. If all the photons reflecting from an object were at 510 nanometers, the object would be highly saturated as green, and it would be this color right here. The eye acts much like a camera. The eye has a transparent sac called the lens that widens and narrows to focus light on the retina. Also leading to proper focusing is the cornea, or the curved outer cover of the eye that changes the light beam as it enters the eye. The shape of the lens is controlled by a set of muscles around the lens called the ciliary muscles. When the lens is made to widen or shorten to allow for focusing, it is known as accommodation. At birth, about 15% of males and 18% of females have severe enough eye problems to require correction. By age 20, until presbyopia, that's where I am, 48% of females and 32% of males require correction. It's almost half of females should be wearing glasses and about a third of males. I was lucky I had perfect eyesight until I was about, I don't know, about 50, 55. Then my eyes started getting presbyopia kicked in, and I was suddenly a little bit nearsighted. Nearsightedness or myopia is where the eyeball is too long and the clearest focus occurs before the light hits the retina. Farsightedness or hyperopia is where the eyeball is too short and the best focus point is past the retina. And astigmatism is a misshapen cornea, elongated, that causes the light to pass incorrectly into the eye and cause the individual to be nearsighted. I had a sister who was myopic, and she was the only one of us that wore glasses when she was in school. None of the rest of us needed glasses, which my parents were really happy about. Anyway, we didn't have to wear glasses. And then my other sister got glasses just before she graduated from high school, I think. Anyway, now everybody wears glasses, I guess. The iris is a muscular covering, of course, we're all in our 70s, and my oldest sister has died. Let me think. Johnny's 70, he's born in 46, so he's getting close to 80. He's 79, will be 79 in September. Nancy's born 48, which makes her 76. I was born in 49, which makes me 75. I'll be 76 in October. Where's Nancy? 77. She's 77. Her birthday's in February. Who else do we have? Russell was born in 51, which makes him 74, or he will be in July. And then David is born in 54, so he'll be 71 in July. Not important. The iris is a muscular covering under the cornea that gives the eye its color. Non-brown eyes were a mutation that appeared about 6 to 10,000 years ago. So before, they're like all dogs and cats have the same color eyes. Well, not humans had the same color eyes until about 6 to 10,000 years ago. The pupil is an opening in the iris that allows light to enter the eye. The movement of the eyeball itself is controlled by the extraocular muscles. The photoreceptor cells of most animals are part of the retina on the back of the inside of the eyeball. The retina is very thin, but still contains 5 distinct layers. The first layer is the optic nerve fiber layer. This is working from the inside out. The second layer are the ganglion cells. The third layer are the bipolar cells. The fourth layer is the actual photoreceptor cells themselves, the rods and the cones. And the fifth layer is made up of pigmented epithelium, and that is what reflects in the dark. Your pigmented epithelium. The rods and cones detect light that reflects off the pigmented epithelium. The rods and cones release neurotransmitters that synapse with the bipolar cells. The bipolar cells synapse with the ganglion cells that form the optic nerve. Now, if we're looking at an animal, the better their eyes, more reflective their eyes are at night, the better their eyesight at night. So the more reflection they have. Why would that be? Well, why would dull eyes not see very well in the dark? Well, the answer is because you're trying to reflect light off of that surface, and the brighter that surface is for very little light, the better you can see at night. So the brighter somebody's eyes are at night, the better they can see at night. And humans, of course, we see all these cats, and here's a cat, and here's a, I'm not sure what that is. I think that's another cat, but this is a deer. The reason that they can see so well is because they have a better reflection. You can see that they're white and green and whatnot. And you can tell when an animal is starting to go blind, like a dog, because their eyes at night will be greener, will be a little bit darker. Humans have red, and that's not very reflective. I'll be back. Okay, let's talk about something else. The rods and cones detect light that reflects off the pigmented epithelium. The rods and cones release neurotransmitters that synapse with the bipolar cells. The bipolar cells synapse with the ganglion cells that form the optic nerve. Well, most of the communication in the retina runs from outer layer to inner layer in a vertical pattern. Amacrine and horizontal cells communicate with the cells in a horizontal pattern. The horizontal cells connect receptor cells. Amacrine cells connect the bipolar and ganglion cells. The rods and the cones represent different functional systems. The scotopic system consists of all the rods in each eye, about 100 million cells. The scotopic system is very sensitive to weak light but cannot detect color. Light is captured in the scotopic system by the photopigment rhodopsin. The scotopic system is located primarily outside of the fovea. So it's not directly, with your direct vision, it's your peripheral vision. The photopic system, so we had the scotopic system, now we have the photopic system. The photopic system consists of all the cones in the eye, about 4 million. So we have 100 million that just detect light, and we have 4 million that detect color. This system detects color and requires relatively strong stimulation, and therefore is primarily a day vision system. The phototopic system is located primarily in the fovea, but is present in other areas of the retina, but in a lower concentration. There are 3 forms of photopigments called opsins that detect the 3 color groupings. The photopigment from the scotopic system is rhodopsin. The photopigment from the photopic system is one of the 3 configurations of opsins. All the photopigments in the eye are made up of 2 parts, retinal and opsin. Retinal is short for vitamin A aldehyde or retin aldehyde, shortened to retinol. Retinol. Light activates the rhodopsin molecule to rapidly lose its retinol, leaving an enzymatic receptor site on the opsin molecule. The enzymatic opsin molecule combines rapidly with G protein transducin. Transducin in turn acts on phosphodiesterase, or PDE, to convert cyclic guanosine monophosphate, cyclic GMP, to 5-GMP. Cyclic GMP inhibits sodium channels, closing with the conversion. The sodium channels close. The loss of sodium ions depolarizes the neurons, and then you see things. Since we are dealing with millions of receptor cells at the same time, weak light will only hyperpolarize some of the receptors, and bright light will hyperpolarize most of the receptors. Thus, the response will always be graduated as to the amount of light that is detected. This form of response leads to sensitivity. It allows for adaptation. It allows the individual to integrate the stimulus over time. There are 2 types of bipolar cells that transmit information, off-center bipolar cells and on-center bipolar cells. On-center bipolar cells are activated when light is turned on. Off-center bipolar cells are activated when light is turned off, so that the eye still has visual capabilities. It detects light coming in from the periphery of the visual field. Photoreceptor cells release the neurotransmitter glutamate. Glutamate is decreased in off-center bipolar cells when light is weak or turned off. Glutamate is decreased in on-center bipolar cells when light is turned on in the middle of the visual field. Each cone in the fovea is connected to 4 types of bipolar cells. 1 on-center midget bipolar cell, 1 off-center midget bipolar cell, several on-center diffuse bipolar cells, several off-center diffuse bipolar cells. Each midget bipolar cell connects to just 1 cone in the fovea. Each diffuse bipolar cell connects to several cones. When light is turned on-center bipolar cells depolarize on-center ganglion cells. When light is turned off-center bipolar cells depolarize off-center ganglion cells. Stimulus only affects the part of the retina that is in the visual field. The retina will create a mosaic of stimulation that the brain puts together as a picture. There are various ways that the eye adjusts to different light intensities. The pupil of the eye will shrink with bright light and widen with dim light. Rods and cones have different thresholds. This is range fractionation. Photoreceptors will adjust their range of reaction depending on the intensity of the light. This will give the individual visual acuity despite the quality of the illumination. Calcium is a major player in photoreceptor light adaptation. When an individual changes from one extreme light to another, it takes time to adapt to the new intensity of light. This is because the retinol and opsin break down at a higher rate in bright light and must regenerate to be available in the dark. Adaptation is much quicker when adjusting from dark to light because the retinol opsin level is not depleted. Color acuity is more accurate in the center of the visual field because the fovea is almost exclusively cones. Moving out from the fovea, the number of cones decreases and the number of rods increases. Light sensitivity is most acute about 20 degrees off center, where the concentration of rods is greatest. About half of the primary visual cortex, V1, is devoted to the fovea. This is one of the reasons that humans are able to concentrate their spatial field on tasks close at hand, such as reading and surgery. A scotoma is a gap in the perceptual field, usually due to injury of the visual mapping area of the brain. People with scotoma will often compensate for their problem by allowing the part of the brain not affected to dominate the affected area. This is known as blind sight. This is what you are actually seeing, and this is what your brain is telling you that you're seeing. Now, if we concentrate on the middle of the red plus, slowly this gray area right here will disappear. Your brain will start filling it in. Well, you guys can come back to this. Each eye is divided into 2 parts, the nerves of the temporal half of the visual field and nerves of the nasal half of the visual field. As the optic nerve leaves each eye, the nerves come together in an area known as the optic chiasm. At the optic chiasm, the nasal optic nerves cross to the opposite hemisphere, and the temporal optic nerves stay on the same side. After the optic chiasm, the nerve pathways are known as the optic tract. Most axons of the optic tract terminate in the lateral geniculate nucleus, or the LGN, of the thalamus. From the LGN, the axons radiate into the primary visual cortex, the V1, of the occipital lobe. The primary visual cortex is sometimes called the striate cortex because of the striations that run through the area. Visual areas of the brain not in the striate cortex are known as extrastriate cortex. Some axons of the optic tract send their axons to the superior colliculus, where the superior colliculus helps the eyes track rapid movement. The LGN is arranged in 6 layers. The outer 4 are the parvocellular layers, and the inner 2 are the magnocellular layers. The P cells have small receptive fields, while the M cells have large receptive fields. M cells are sensitive to low contrast and rapid change. About 10% of the retinal ganglion cells are M cells. 80% of the retinal ganglion cells are P cells. P cells require high contrast to be stimulated. P cells are relatively insensitive to rapid temporal change, such as flicker. P cells are sensitive to different wavelengths of light, and they therefore detect color. P cells have sustained responses. P cells have high acuity. The primary visual cortex is the first and most abundant area in the human brain. From the V1, the stimulus is farmed out to other areas of the brain, depending on what part of the eye the stimulus comes from. Areas are designated from V1 to V5. Researchers have discovered that there are 3 different kinds of cones. Cones that are sensitive to short wavelengths only, cones that are sensitive to long wavelengths only, and cones that are sensitive to the middle wavelengths. Of the middle wavelength cones, some are more sensitive to the higher ranges of middle. Some of the middle range cones are more sensitive to the lower ranges of middle. There are twice as many long wavelength cones, reds, as middle wavelength cones, greens and yellows. Short wavelength cones are the fewest, blue and purple. This may be one of the reasons why so many people think red is their favorite color. Myopia is a condition where the eyeball is elongated, causing the image to focus on the front of the retina, causing a blurred image. There is some evidence that farsightedness is caused when children look at images too close to their face. And this is one of the reasons why they tell children not to sit too close to the television set. This can be prevented by only allowing reading in adequate light, avoiding small type, reading without glasses anytime that you can do so. Glaucoma is a group of eye conditions that damage the optic nerve, the health of which is vital for good vision. This damage is often caused by an abnormally high pressure in your eye. Glaucoma is one of the leading causes of blindness for people over the age of 60, and my brother does have glaucoma, and he has to put eye drops in his eye to lower the pressure every day. And that is that. So let's go ahead and get rid of this one, and we'll go to motor control and plasticity. One of my favorite chapters. Muscles cover the skeletal system and provide movement. They also provide protection for the organs that lie underneath. How muscles attach to the bones is an indication of the type of movement that they perform. Muscles only provide force when they contract, tighten up, get smaller. So when you're making a bicep, when you're trying to show everybody how big your bicep is, what you're doing is you're shortening it. If you have it stretched out, your arm stretched out, then your tricep is actually being contracted. But if you move your arm in and make a 90-degree angle, then you're showing off your bicep. Some muscles maintain body posture. Some muscles produce movement around a joint. Any movement that occurs in the body is performed by muscles. Muscles are connected to bone by tendons. Around a joint, there are 2 sets of muscle groups. When one group contracts, the other extends. These muscles are known as antagonists, like your biceps and your triceps. Your triceps are on the back of your arm. Your biceps are on the front of your arm. Muscle groups that act together to perform the same act are known as synergists. Muscles are made of thousands of fibers. These muscle fibers are made up of two kinds of filaments, thick and thin filaments arranged in a regular pattern. Thick filaments are made up of myosin, and the thin filaments are made up of actin. There are two basic types of muscles in the human body. Smooth muscles contract in an automatic fashion without human control, regulated by the autonomic nervous system. Heart muscles and muscles in the digestive tract are examples. The other type of muscles are striated muscles, primarily supporting the skeletal system, and you can see that they are striated. Since different muscles perform different tasks, each requires different speeds, precision, strength, and endurance. Striated muscle fibers come in two forms: fast muscle fibers and slow muscle fibers. Eye movement must be quick to follow the movement of speeding objects, but only for short periods of time. Extraocular muscles are fast muscle fibers. Fast muscle fibers react promptly and strongly. They fatigue very rapidly. They're used when muscle tension must change frequently. Slow muscle fibers have greater resistance to fatigue and are used mainly to maintain your posture. Muscles react to stimulus coming in from motor neurons. The axon will split into many branches and activate many muscle fibers. As the axon is activated, it releases the neurotransmitter acetylcholine. Acetylcholine makes the muscles react by opening sodium and calcium channels in the muscle fibers. The ionic change allows the muscles to contract. The point where the muscle fiber and nerve structure communicates is the neuromuscular junction. Neuromuscular junctions are large and very effective. And that's a neuromuscular junction right there. Each neuromuscular junction releases enough acetylcholine to not only cause one reaction, but several at the same time. One cause of muscle fatigue is due to diminished effectiveness of the neuromuscular junction. Muscles develop coordination when axon terminals migrate to specific target muscles, and acetylcholine receptors migrate to the clustered axon tips, allowing for more efficient muscle reaction. Muscles react because spinal or cranial nerves send messages to the muscles through efferent neurons. Motor cells of the spinal cord are larger and faster than other motor neurons. Generally, small motor neurons activate slow muscles and are more easily excited. Large motor neurons tend to activate large muscles, which require more stimulation for activation. And I'm going to show you some right now.
The skeletal muscle of animals, including humans, are striated. They exist in bundles of thousands of muscle fibers. A single muscle fiber is actually a muscle cell. As you can see, muscle cells are very elongated and contain several nuclei. Packed inside the muscle cells are contractile myofibrils. Myofibrils show the striated pattern as repeating subunits called sarcomeres. When a motor neuron stimulates a muscle fiber, overlapping filaments in the sarcomere slide along one another, and the sarcomeres shorten. The repeated pattern of the sarcomere results from the arrangement of the two major proteins within the myofibrils: myosin, and actin. The myosin heads are the actual mechanism for muscle contraction. Now let's examine the process in detail. First of all, when a motor neuron stimulates a muscle fiber, calcium ions are released from the sarcoplasmic reticulum. The ions force the tropomyosin to displace and expose myosin binding sites on the actin. An ATP molecule is then hydrolyzed, and the myosin head forms a cross bridge. When the ADP is replaced by a new molecule of ATP, the myosin head detaches from actin and goes back to its original state. And that's pretty much the whole process. And one more thing. Each myosin head attaches to and releases from actin independently, not in unison. Otherwise, the myosin heads would all release at once, and the whole muscle would go to a relaxed state. And that wouldn't be good.
Wow. You dropped the weight on your foot. How funny. The body must maintain control over the position of its parts. This knowledge of the position and state of its muscles and limbs is called proprioception. This knowledge is maintained by select receptors that report muscle and joint positioning. Muscle spindles lie parallel to muscle fibers. Golgi tendon organs report on the position of the joints and are situated with one end attached to the tendon and one end attached to the muscle. Muscle spindles are located in many areas of our skeletal muscular fiber. Muscle spindles are actually spindle-shaped. It consists of an afferent element and an efferent element. Their primary function is to maintain your posture and tell you when you have been in a position too long. When a muscle fiber is activated, the muscle spindle reports rate of change. Rate of change refers to the change the fiber has gone through to be able to function. The muscle spindle also reports force, the force required to maintain contraction. Muscle spindles have two types of receptor endings. Primary sensory endings, wrapped around the middle of the fiber, are activated during the initial phase of contraction and then adapt to a lower discharge rate. Secondary sensory endings are found near the smaller ends of the fibers. They are not activated at first, but only after prolonged use, and they tell you when you're tired. Muscle spindles are informed of impending action through two special motor neurons. Gamma motor neurons, or gamma efferents, inform of change of position from the initial movement and comprises about 30% of the efferent fibers. Alpha motor neurons perform initial movement. 70% of the efferent neuron fibers are alpha motor neurons. While individual muscle movement is monitored by the muscle spindle, muscle contraction is monitored by the Golgi tendon organ. Golgi tendon organs are able to monitor muscle and joint movement because joint movement is controlled by tendons connected to muscles. And there you go, that's what a Golgi tendon organ looks like. Golgi tendon organs are somewhat insensitive to passive muscle stretch because they are connected to the elastic component of the tendon, and it is only activated under heavy loads. The Golgi tendon organs detect overloads that threaten to tear muscles and tendons. Activating the Golgi tendon organs inhibits the motor neurons supplying the muscles that pull on the tendon, and thus by relaxing the tension prevents mechanical damage. And now I'm going to show you what muscle building looks like.
Muscle building. The key to building muscle is protein synthesis and cellular repair. Skeletal muscle is made up of long fibrous chains containing proteins. Bundles of thousands of fibers make up the muscle itself. When an athlete engages in a heavy workout, this structure is damaged, causing micro tears in the muscle fibers. The body naturally repairs this damage by mending the torn fibers. The result is a muscle that's larger and stronger than before. And with each additional workout, the athlete adds bulk, strength, and speed.
There you go. What was I going to say? They were talking about building muscle. Oh, I was talking to Dr. Barber the other day, and I was asking him what he eats before he exercises. Both of us are lift weights, and he was saying that he eats 3 eggs, and that's exactly what I eat. I do. Always eat eggs before I go work out. I never eat eggs any other time. I just eat eggs when I work out because it's almost pure protein, and that's what I'm looking for. I'm trying to build muscle. So Dr. Barber and I are on the same page. Let's try this one. I think it still works. Yes.
Ready to tackle your debt? We can help. A Stretch Financial Wellness Vision subscription can help you visualize your debt picture. Muscles. We have over 600 of them. They make up between 1/3 and 1/2 of our body weight. And along with connective tissue, they bind us together, hold us up, and help us move. And whether or not bodybuilding is your hobby, muscles need your constant attention because the way you treat them on a daily basis determines whether they will wither or grow. Say you're standing in front of a door, ready to pull it open. Your brain and muscles are perfectly poised to help you achieve this goal. First, your brain sends a signal to motor neurons inside your arm. When they receive this message, they fire, causing muscles to contract and relax, which pull on the bones in your arm and generate the needed movement. The bigger the challenge becomes, the bigger the brain's signal grows, and the more motor units it rallies to help you achieve your task. But what if the door is made of solid iron? At this point, your arm muscles alone won't be able to generate enough tension to pull it open, so your brain appeals to other muscles for help. You plant your feet, tighten your belly, and tense your back, generating enough force to yank it open. Your nervous system has just leveraged the resources you already have, other muscles, to meet the demand. While all this is happening, your muscle fibers undergo another kind of cellular change. As you expose them to stress, they experience microscopic damage, which in this context is a good thing. In response, the injured cells release inflammatory molecules called cytokines that activate the immune system to repair the injury. This is when the muscle-building magic happens. The greater the damage to the muscle tissue, the more your body will need to repair itself. The resulting cycle of damage and repair eventually makes muscles bigger and stronger as they adapt to progressively greater demands. Since our bodies have already adapted to most everyday activities, those generally don't produce enough stress to stimulate new muscle growth. So to build new muscle, a process called hypertrophy, our cells need to be exposed to higher workloads than they are used to. In fact, if you don't continuously expose your muscles to some resistance, they will shrink, a process known as muscular atrophy. In contrast, exposing the muscle to a high degree of tension, especially while the muscle is lengthening, also called an eccentric contraction, generates effective conditions for new growth. However, muscles rely on more than just activity to grow. Without proper nutrition, hormones, and rest, your body would never be able to repair damaged muscle fibers. Protein in our diet preserves muscle mass by providing the building blocks for new tissue in the form of amino acids. Adequate protein intake, along with naturally occurring hormones like insulin-like growth factor and testosterone, help shift the body into a state where tissue is repaired and grown. This vital repair process mainly occurs when we're resting, especially at night while sleeping. Gender and age affect this repair mechanism, which is why young men with more testosterone have a leg up in the muscle building game. Genetic factors also play a role in one's ability to grow muscle. Some people have more robust immune reactions to muscle damage and are better able to repair and replace damaged muscle fibers, increasing their muscle building potential. The body responds to the demands you place on it. If you tear your muscles up, eat right, rest, and repeat, you'll create the conditions to make your muscles as big and strong as possible. It is with muscles as it is with life. Meaningful growth requires challenge and stress.
Ta-da! There you go. And that's why I lift twice a week to maintain my muscle mass. At 75, I'm about the same size I was when I was 55. My muscle mass, anyway. There are two reflexes that are maintained by the spinal cord that are automatic and are not mediated by the brain. The flexion reflex is activated when pain is applied to the leg. The muscles will contract, moving the affected part away from the stimulus. And let me show you some of the reflexes. I think that's what we're going to see. Oh.
The Hulk is an anthropomorphic exoskeleton which mimics the human form. It provides extra support that enables a person to carry more weight than they normally could. Exoskeleton technologies are considered part of the robotics field. If you want to think about them as wearable robots, that's pretty accurate. They sense what the user wants to do, where the user wants to go, it mimics the motion. We like to call these mantonomous systems. The exoskeleton is primarily composed of titanium components, which are both lightweight and provide the strength needed to carry the load. A microprocessor takes in readings from the sensors that are throughout the structure and calculates where the user wants to move, and then commands the actual hydraulic system to actuate the joints to provide the motion. You can walk, you can run, you can bend, you can crawl, you can leap. It does not impede your range of motion whatsoever. This technology could be a real benefit to the military. Just imagine you're a soldier operating at 6,000 feet in the Afghan mountains and being asked to take 120 pounds up in that level of thin air. How exhausted you would be once you got there. An exoskeleton provides the ability to carry that weight the same distance, but to have energy left to execute the mission once you're there. We've also looked at attachments that allow users to carry weight not only on their back, but also on the front. The exoskeleton will fit anybody between 5'4" and 6'2". The current system weighs 53 pounds, but it's pretty much unnoticeable to the user because what the technology does is it even transfers its own weight to the ground, in addition to the weight you're carrying. And we're taking the load off the soldier's back, and we're going to help prevent injuries that occur from carrying heavy loads and repetitive lifting tasks. The best part about working on this technology is that it gives us a glimpse into the art of the possible.
Wow. Okay. So I'll tell you what. Let's see if let's see if they still have this. This is still operational. Hulk. What else? Human Universal Load Carrier, NASA, being developed by NASA. Army Technology. This is in 2020. I think that's when I put it in there. There we go. Same stuff that we just saw. So it looks like, I don't know if it's operational now or not. This is just getting, let's type in there something about that. Stuff about it was never fielded. Okay, so it's not been. It was never fielded or deployed with combat troops. So it was tested by the United States Army, but it's not being used. Okay, well, it's just a thought. In knee-jerk response, the quadricep muscle that is contracted to allow the knee to bend is informed of tendon pressure that makes the muscle react to stretching, thus the stretch reflex. So we've all done this. They've tapped our knee right below our kneecap, and our foot has kicked out. The primary lateral cortex, the M1, controls all the muscle movement in the body. The hands and lips utilize the biggest portion of the area compared to the percentage of the body that they represent. The M1 communicates with the rest of the body through the pyramidal system. The pyramidal system is a triangular-shaped bundle of nerves that runs from the brain through the medulla and into the spinal cord. Lesions to this system will result in the inability to move select joints and limbs. Those neurons that run from the M1 to the muscles that are not in the pyramidal system are said to be in the extrapyramidal system. These neurons control the spinal reflexes. And now we're going to see all the spinal reflexes. Maybe.
Honestly, no one wants to think about HIV, but there are things that everyone can do to help prevent it.
Yeah,
There
we go.
The lower body reflexes will be performed in the same manner as the muscle strength exams were performed. We're going to test one side and then quickly test the other side and compare side-to-side responses. We've got 5 reflexes that we're going to go over, one being the knee reflex, one the Achilles reflex, and then the adductor reflex, the internal and external hamstrings. The first one will be the knee reflex. I'm going to have the patient close their eyes, strike the tendon. We got a good response. I want you to keep your eyes closed. Strike it again. Got another good response. Now the Achilles reflex. I'm going to just slightly dorsiflex the foot by reaching underneath the foot and letting her rest the foot on my hand. And then I'm going to reach back and I'm going to tap the Achilles tendon. And I got a real nice plantar flexion of the foot. Comparing side to side, I have the patient lay face down. Now we can do the adductor, internal and external hamstring all in this position. If I'm going to test the adductor, I'm going to have the patient medially rotate the leg. I find the adductor tendon along the medial side of the leg. I've located it, and I'm going to strike over the tendon, and I get a slight contraction of the adductor. For the internal and external hamstring, I just have the patient lay flat. I locate the tendon by slightly having the patient bend their knee against resistance, bring the leg up. I'm going to push down. And the tendons pop out quite easily. I've got them, you can relax the leg. I've got the tendon located, the medial hamstring tendon. I'm going to strike the tendon, and I'm watching for contraction and slight flexion of the knee. On the lateral hamstring, I locate the tendon, and I'm going to do the same thing, strike the tendon, and watch for contraction of the hamstring as well as slight flexion of the knee.
Ta-da! There you go, all the spinal reflexes. The extrapyramidal system communicates with the spinal cord through two tracts, the reticulospinal tract and the rubrospinal tract. The extrapyramidal system not only handles spinal reflexes, but muscles responsible for breathing and muscles that control the head and the neck. And that is the end of chapter 11. So I'll talk to you guys next week. We'll tackle these chapters.